I'm about to copy. Okay. Okay. Protease. So protease convert proteins into amino acids. Okay, the next one in experiment was set up to investigate the effect of enzyme on a coagulated mean the clotted. Okay. Albumin is the white part. Okay. It means protein. Okay, an experiment has been done. So in first one we have added the dilute acid. In the second one we have added enzyme with dilute alkali. In third one we have added water. While the fourth and fifth one they contain only acid and alkali. Okay. So where do you think in the digestive system is this process take place? Okay, this one is quite easy. Think again. Where is protein digested? Where is protein digested? It's in the stomach. Wait. Protein is digested by pepsin, right? Where is pepsin produced? In the stomach. So answer would be D. Okay. Now question number 23. I guess we have done this. Or is the question similar to we did? I guess. Similar. Okay. Dish is filled with agar jelly, and we have made certain holes. And in holes, there are one contains pepsin, the other contains distilled water, then one contains the agar jelly containing protein, and then hydrochloric acid and the boiled pepsin solution. So after 30 minutes, which hole is surrounded by the largest area with protein? You have to tell only one. A. Think again. No. Think again. What conditions are? Yeah, without protein. Look, look. No, no, still no. Wait. Without protein means pepsin is going to act on that. But for pepsin, we need some condition. What is that condition? The acidic condition. So answer would be B. Okay, wait. We say that proteins are digested by pepsin, okay, and it's present in stomach. What are the conditions of stomach? The acidic one. It means pepsins work best at acidic condition. Now, if they have asked you about without protein, it means all the protein has been digested. So which condition is the best? B. No, it would be distilled water. It would be distilled water because there won't be any enzyme present. Okay. Only two of the following statements accurately describe what happened in the mouth. Number one, amylase break down starch molecule into smaller maltose molecule. Yes. Chewing increasing the surface area. Yes. Saliva emulsify fat. No. Saliva only bind the molecule. Teeth break up large molecules into smaller soluble molecules. No, they are not soluble. They are still insoluble. So which statements are correct? 1 and 2. Which statements about enzyme is essential to the lock and key hypothesis? Enzyme molecules, sorry, enzyme molecules have distinct shape. Yes, it's more related to lock and key model. Okay, 27th one, there is a lock and key model given, different shapes are given. Let's have a look on those shapes. The diagram represent the enzyme molecule um, and its substrate, which diagram shows that it has been heated to 100 degrees Celsius, means the enzyme got denatured. So which one is correct? C. Good. Which chemical shows the presence of enzyme in a biological powder? Okay, what is an enzyme and what test we are going to use? Yeah, enzyme is a type of what? good. It's a type of protein and we have to use butyrate. Okay, next one. Again, a graph is given. So which graph shows the reaction in the alimentary canal? Means it's related to human. So which graph is the most appropriate one? C, good. Now the next one is there are three diagrams given. Starch to maltose and okay, what is Q, what is R, and what is S in these diagrams? P is okay, okay. Starch is Q. Okay. R no, no, no. Amylase is an enzyme. Which is an enzyme? P. Enzyme is P. R and S are products. Exactly. Starch, Q. So the only correct option is A. Okay. I guess we have done the 30th one, the amylase one. Okay, let's move to the 31st then. Wait, let me check. Yes. No, no, starch works best at at the neutral pH. It would be A. It won't be B. Okay, if the normal is given, so here they have mentioned it's acidic. It means this one is not acidic. So it must be neutral. Okay. Okay, the graph shows the effect of temperature on chemical reaction. At what point most product molecules being released? Where are the most products formed? C. Good. Okay. So this is all for this paper. Let's discuss the other paper. Okay. I'm not discussing movement across the membrane. Let's discuss the enzymes because we are discussing. Let's finish it up.